aquaculture, fish are caged, and these will create unsuitable environment for the production of fish. What can we do to increase the reproduction of fish? Let's check out our research.
So for this project, we will use Nile tilapia, or its scientific name Oreochromis muloticus. This fish is actually a very famous cultured fish, whether in Malaysia or abroad. And there are many studies about this fish, but there is less studies for the reproductive system like ours.
So what is PCR? PCR stands for polymerase chain reaction. It is originally developed in 1983 by the American biochemist Carrie Mullis. It is used to amplify a few small amounts of DNA to a millionth copy of specific DNA sequences.
Gonadotropin is a critical modulator of gametogenesis and gonadal spermatogenesis in fish. It has two main types of hormone. There is the luteinizing hormone and the follicle stimulating hormone. These hormones involved in the reproductive system of fish and mammals.
Our objectives are to detect gonadotropin hormones, which are follicle stimulating hormones, and luteinizing hormones of tilapia by using PCR assay. And we also wanted to analyze gonadotropin hormone gene sequence by using blast nucleotide or BLAST-N.
First, we dissect the fish and cut its head using these dissecting tools to collect pituitary, which is located behind the brain. For pituitary contain gonadotropin hormone, which is the starting key for our research. Then, we proceed to DNA extraction. Then, we perform PCR amplification. So these are the ingredients. There are DNA template, buffer, primer, and others. Next, we run the gel electrophoresis to detect the gonadotropin hormone. Then, we send the PCR product for DNA sequencing. And lastly, we use blast nucleotide to analyze the sequence. So, for the results, we will use the blast nucleotide or the BLAST-N to compare the DNA sequence that we got from the PCR together with the DNA sequence that is in the gene bank. And for your information, the gene bank is actually a place which stores information on DNA sequences and genetics, not only for human beings but also ranging from bacteria and as normal human beings. So for our research, we use the follicle stimulating hormone gene and also the luteinizing hormone gene. For the follicle stimulating hormone, we're able to receive the result of 310 base pair, and for the luteinizing hormone gene, we're able to obtain the result of 450 base pair. And thankfully, we were able to successfully match our result together with the DNA sequence in the gene bank.
So, through this research, we will obtain tilapia hormones, which can be used to any kind of fish. This hormone will increase their rate of reproduction. Hence, it will increase the rate of producing in aquaculture in fishery. So, this will increase the national income in aquaculture.
Next is the impacts and innovation. For our research, we were able to obtain 3 impacts. The first is the detection of gonadotropin hormone, which is the follicle stimulating hormone, where we obtain the result of 310 base pair, and also the luteinizing hormone, where we obtain the result of 450 base pair. The second is the DNA sequence analysis from the blast end, where our PCR bands in our research were detected as follicle stimulating hormone and also the luteinizing hormone. And the third impact is the polymerase chain reaction, which is the PCR. Why? Because the PCR was able to give a rapid results because it's very sensitive to assay, and also the PCR was able to detect hormone and genetics from fish and also other living beings.
As in the aspect of commercialization potential or marketability, we are using the PCR assay to measure the gonadotropin level in tilapia and other pisciforms. We could also develop an artificial hormone-based tilapia DNA sequences and breed other fish.